So the next speaker, we actually have Rudy the Rhino making a plea for the world. I actually see Rudy in the audience, but he is not going to identify himself until after this session. And I'm going to introduce and bring up to the podium Jeanne Loring. Jeanne, where are you? Let's see. Oh, come on up. So my colleague Jeanne Loring, besides having been honored with the Stem Cell Action Award by the World Stem Cell Summit and Regenerative Medicine Foundation, is truly a pioneer in the field of stem cells and pluripotent stem cells. She works very closely with one of our favorite nonprofit groups called the Summit for Stem Cell. That has no affiliation with the World Stem Cell Summit, but is a patient advocacy organization related to Parkinson's disease. She's an entrepreneur and having created through her science, Aspen Neuroscience, professor emeritus at Scripps Research. But let me tell you a little bit how I described Jeanne at a recent presentation, because while she has a Rosalind Franklin Award and other numerous awards, and was considered one of the top 11 most influential women in advanced therapies. Does that mean you're only 11th? I know. Anyway, she's interested in the development of broad applications of pluripotent stem cells, described as a stem cell evangelist and the godmother of stem cells. I think of what Jeanne Loring's career has been as an odyssey, as a true odyssey. And every odyssey needs a hero. And Jeanne Loring is one of my heroes. I introduce my friend, Jeanne Loring.
Thank you. Rudy's first. I'm just going to watch.
Oh, first Rudy.
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 You do this. Oh, you're doing
And
this.
then I'll get this.
You know, Bernie, this jacket and tie is really uncomfortable. You know, it took me 2 days to get this tie done right. And oh, wait a minute. Are we recording? My name is Rudy the Rhino. On behalf of rhinoceri everywhere, I want to thank Bernie Siegel and the World Stem Cell Summit for providing me this platform to make a brief statement. You know, my cousins, the northern white rhinos, are functionally extinct. Sad but true. Probably they were hunted and poached for our horns. Maybe it's the whole Anthropocene extinction thing they're talking about. Anyway, what can any of us do? But now, thanks to stem cell research and the discovery of iPSCs, plus the work of brilliant scientists like Dr. Jeanne Loring, there's hope. Believe me, this is important for more than just us odd-toed ungulates, but all living things, especially mammals like you crazy Homo sapiens. Dr. Loring will tell you all about it. Please listen and think about supporting the Frozen Zoo and other meritorious nonprofit groups dedicated to saving our planet. Now, somebody please help me get out of this tie.
That was one of the best introductions I've ever had, I have to say. It's going to be hard to top that one. So I'm going to talk about one of the stem cell projects I've been working on for more than the last 10 years. And that is the idea that you can use stem cells to rescue endangered species. My partners in this work are Oliver Ryder, Marissa Carodi, who are at the San Diego Zoo Global. They've been working very hard. The work is now going on in their lab at the zoo. And if you ever in San Diego, I highly recommend that you take the tour and see some of these rhinos who are going to be. Yeah, there's Nola. So Nola was the last one of the northern white rhinos who was at the San Diego Zoo. She died in 2015, and now there are only two, as Rudy the Rhino said, only two left, and they're non-reproductive females. They're in a protected reserve in Kenya. And the reason why we're working on this project is because of Oliver and Marlis, who are the directors of what is called the Frozen Zoo at the Conservation Research Institute at San Diego Safari Park. They've been freezing cells from animals that have died or animals that are endangered ever since the 70s. And they didn't know at that time what they were going to do with them. And it actually wasn't until iPS cells were invented by Shinya Yamanaka that it became obvious there really was something we could do with them. This is the Frozen Zoo. And this is you probably already know this, but this is the breakthrough that led to the ability to make iPS cells from anyone. It started, of course, with humans. The original stem cells were made from, these are pluripotent stem cells. They have two qualities which I have to keep telling people about. They can live forever. They can keep dividing forever. The only cells that aren't cancerous that can live forever and keep dividing. And they have this additional quality, which is they can make everything in the body. And interestingly, even gametes like sperm and eggs. So this technology has opened up the door to some really imaginative approaches, including conservation. This is how it started. It was 2008, and I had just started at Scripps Research Institute. I'd moved from the Burnham Institute to Scripps, and I was founding what is called the Scripps Research Center for Regenerative Medicine. And I knew Ollie Ryder, from some meetings that we'd gone to, and so it's not very far from Scripps Research to the Safari Park, from Scripps to the Safari Park. It's about a half-an-hour drive if the traffic's not too bad. And so I took my entire lab out for a celebration at the zoo, at the Safari Park. We took a safari, which is we get on this truck, we go around, we feed animals. And one of the people who was on that, in that celebration was my new postdoc named Inbar Friedrich Ben Nunn. Inbar went on the next few years to focus all of her effort on reprogramming something other than humans and mice. And the animals that were chosen by Ollie were the northern white rhino and the drill. The drill is a primate, very pretty primate, but also endangered. And she adapted the methods that have been first used for mice and then for humans to reprogram the first cells ever from any endangered species, and in fact, any species other than mouse and humans. And we had, it was amazing because when we came back from the safari, I had my whole lab around me and I said, does anybody want to take this on? And she just ran up to me and said, please let me do this as a project. And she's a new postdoc, so in theory, this is her whole career ahead of her. This is kind of a chancy thing. What if it doesn't work? But she embraced it, and she did a fabulous job of getting it to work. So we had the paper published in Nature Methods. We had the cover of the paper, this imaginative cover that shows all the animals in this ark being rescued by pluripotent stem cells. And in 2015, there was a virtual reality documentary made about the northern white rhino, and I'm lucky enough to be part of it. It's called The Ark, and it was made by Keel and Eileen over a number of weeks. It premiered at the Tribeca Film Festival in 2016, and you can now get it apparently on Hulu. I do not have an Oculus. I cannot get it, but maybe you can. Other people have. The picture is during the filming, and the person standing up in the very back is Ollie, and the one right next to him in the hat is me. And that's Nola. Also in 2015, we really started getting getting a little bit of traction because other people in the world, because of our paper, thought it might be interesting to start using methods like this to conserve endangered species. So there was a meeting that was called in Vienna in 2015 and included people who were conservation people, genomics, reproductive physiology, and us, the stem cell scientists. And this is a picture of us. Again, Ollie is in the front. I'm right next to him. And the woman in the red shirt is the amazing reproductive physiologist, Barbara Duran. This is our plan. This was published in 2016 in the journal Zoo Biology, which was the first for me. I'd never published in that journal before. And it made quite an impact on this particular field. And this is our outline for how we could rescue the northern white rhino. And actually a lot of this has happened. The two animals who are in Kenya have had their oocytes collected, and there's been work done in Germany by Tom Hildebrand's lab to essentially make embryonic stem cell lines from these. And in our lab, we started making iPS cells, and our hope is to make those iPS cells into sperms and eggs, sperm and eggs, and to do in vitro fertilization to generate new rhinos. And in order to do that, you need to have more than one animal. If you have one animal, you can just clone it forever, but that does not rescue a species. So we took the 12 frozen samples of skin and reprogrammed 9 of them of the northern white rhino samples and 2 of the closely related southern white rhino. This was work that was done almost entirely by Marissa Carodi, who worked in my lab for a number of years and is now full-time at the zoo. She has her own lab there. And this paper was published in Stem Cells in Development with a picture of Noah on the cover, which was really close to my heart. Sorry, close to my microphone. So now we have enough iPS cell lines to theoretically have enough genetic diversity in order to create and rescue the species, not just an animal, but the entire species. And I think, that's not working, is it? This is really important. Okay, well that thing you see in the center there are beating heart cells. And if you could see them, you'd be really impressed because they come from an animal named Angelifu, who has been dead for many years. We made his iPS cells and we turned them into beating heart cells in a culture dish. And we've turned them into neurons, we've turned them into a lot of other things. I don't know where you can show the movie from, but it's not beating though, it's not moving, is it? Oops, sorry. No. Nope. What should I do? We have plenty of time. I'll let him take over. Oh, yeah. Whoa. Thank you. Okay, so now you can tell why I'm so impressed. I'm taken by this. So these are our heart cells from an animal that's been dead for at that time, about 7 years, and is one of the last of his kind. In fact, he was one of the last males that existed. So our iPS cells are capable of doing a lot of things. So now I'm going to give you an update on what we're doing now. So what we need to do before we could go much further was to try to bring the field of endangered species iPS cells up to the level of human iPS cells. And in order to do that, we needed to do the equivalent of the Human Genome Project, but with rhinos. And we have now, are just about to publish the annotated genome of the northern white rhino. And we need all these, we need to know about the genome so that we can develop the tools for assisted reproduction. Everything depends on us being able to do genomic analysis of these cells and making sure that they haven't changed in culture, for example. And this paper was published in bioRxives and hopefully will be published in a peer-reviewed journal very soon. So the other thing that's going on is that we are now working on making gametes from these 9 iPS cell lines. And we know this is going to work because it works in mice. And it may take a while, but everything that works in mice, well, everything like this that works in mice, it'll work in another animal, or it'll clearly not work. And in this case, we think it will work because the reprogramming methods worked, and the differentiation methods all work. And so the rhino is similar enough to the humans that we think we can do this. It's similar enough to the mouse that we think we can do this. So this is a paper published just a couple of months ago that shows that not only can you make mice out of iPS cell-derived oocytes, but you can make them from oocytes made from male mice. So that means we can make female gametes from the male rhinos and then have them also make sperm and then do all the intercrosses that are necessary to increase the genetic diversity. I know there are some of you are looking rather startled, but we think it's possible. It'll probably be possible in humans too, so watch out. Now, once we can make an embryo, we're going to be in the hands of Barbara Durant, who is the reproductive biologist. The zoo brought in 6 southern white rhinos to be surrogate mothers for the embryos that we create of northern white rhinos. Their names are Victoria, Amani, Wallace, Helene, Livia, and Nikita. And Victoria and Amani have both had babies by artificial insemination, which is the thing you do before you do in vitro fertilization and transfer of an egg. So that's been accomplished. There is a Rhino Rescue Center at the San Diego Safari Park. You'd be happy to visit or donate if you can't get there. Here's a picture in the upper right of Victoria and her baby Edward, and then Wallace and me. I want to acknowledge at least some of the people who have done this kind of work. There's a group in Kiel in Germany, Franz Josef Mueller, who is my former postdoc, is now a professor, has been for a long time, and he jumped into this project to do the genome sequencing along with Bjorn, who is his postdoc. And at the Institute for Conservation Research, Marisa, of course, is the main leader of that program, along with these other people who have joined her. There's a picture there of Oliver Ryder and me on the truck discussing, I don't know, pluripotent stem cells. And then my entire lab at Scripps Research, because they were doing iPS cells on other things like humans, they provided all the background and trained Marisa in the methods that we had developed for making iPS cells and differentiating them. And I'm going to close with a haiku from my favorite poet, ChatGPT. This is a haiku for the future of the northern white rhino. It says, "Gentle giants roam. Stem cells bring hope for their lives. Rhinos saved, reborn." He's such a better poet than I could ever be. Thank you.
First, I want to introduce my colleague, Kevin McMahon, who is the voice and image of Rudy the Rhino. Please, Kevin. He is the best graphic designer. Stand up, turn around. Rudy, you know, when we save the first rhinoceros, northern white rhino, I'm going to be sure to get you two together. Now, I have a question for you, Jeanne. We've all heard about the company Colossal and bringing back the woolly mammoth, and it seems like the techniques that you are using just might be able to be used, and we can bring back a whole menagerie of extinct mammals. So are you going to try that? What do you think about that? Is that on the horizon?
So let me give you the difference between a woolly mammoth and a northern white rhino is that we don't have any woolly mammoth skin cells in the frozen zoo. If we did, we could do it. The iPS cell work depends on the existence of cells that we can reprogram. Without that, you can get the whole genome sequence, but there's another step you have to make in order to put that sequence into the nucleus of a cell and then turn that cell into a gamete. And that's not my cup of tea. I mean, I'm really good at stem cell stuff. This is going to be very, very hard. So I think we will have a menagerie, but there are so many animals that are, there are 10,000 animals in the frozen zoo, and we've just started getting the endangered ones to, we're reprogramming them. In fact, we're going to have the first meeting of iPS cell technology in San Diego in the fall from people all over the world who have their own species that they are reprogramming, from Australia, from Singapore, from other places in the world. And we're all going to get together and talk about technology. We're going to be we're not going to keep any secrets because there's no point. But I hope that means it jumpstarts this whole field so it becomes easier. I was just in Singapore and I talked to a graduate student and a postdoc who are coming to that meeting, and they just peppered me with questions. They're trying to reprogram cats, and they said, why are cats so difficult? I go, I don't know, I never tried a cat. So we're going to figure all this out. This is the kind of thing that it's going to take teamwork.
Well, we definitely don't have enough cats.
No.
So in our audience is my friend Robert Jacobson, who is an environmentalist as well as an expert on space technology, but he has been interested in actually saving orcas. And have you thought about marine mammals? And, and how, can you explain a little bit what's going on in the marine mammal world?
Yeah, so actually we've reprogrammed, we reprogrammed the Lake Baikal seal with a visiting postdoc from Siberia. So we have those in our freezer. They haven't been characterized yet, but there's no reason to think it's not going to work. And really what it takes is passion for that particular species and the interest to show up in the place where you can learn the technology. Yeah. I'm sorry? Oh, no, yeah, they're on the list. They're on the list.
But have they gathered any samples?
Yes, there are samples, yes. The frozen zoo is also expanding all over the world. So one of their missions right now is to create frozen zoos in other countries so that they can exchange samples and collect them locally. It's not easy to collect a sample in Africa and have it, because you culture the fibroblasts and then you freeze them down. And the technology isn't available everywhere. But certainly in Mexico, yes, yeah.
Alan Giacomo has a question?
Yeah, two questions. First, you made a very tantalizing reference to doing the work. You made a very tantalizing comment about doing this work with humans in terms of the gametes from a single gender. So that entails all sorts of ethical type questions of the sort that Graham was covering.
Oh, absolutely.
And it would be interesting to speak about that. And then the second question has to do with the connection of this work to the work that you're doing on the space station.
Yeah, okay, so the first one. Yeah, the ethical, I always felt that if we could do this in a rhino, then that would sort of pave the way for doing it in humans. And there's actually a company, apparently there's two companies. I know of one in the San Francisco Bay Area that is actually, it's actually being funded by the CEO of OpenAI in order to make gametes from individual humans so that they can, same-sex parents can have genetic offspring. And like I said for everything else, I'm one of their scientific advisors, so that's my disclosure, because it's such cool work. I like to advise people who are doing really cool work. So I think that's on the horizon. I don't know how far out the horizon is. And then your second question was, and you have to remind me, sorry, I have a, oh, space, right. So one of our other projects is sending cells into space, which we'll be talking about in a few days. And it is relevant because we use the same technologies for all of these projects. In space, we're not sending up rhino cells. I want to someday, but that was a little bit over there. They didn't get excited about it, let me put it that way. We are sending up human cells, and we're sending up iPS cell-derived neurons. I would love to send up iPS cell-derived rhino neurons into space and see how they differ from humans. Someday. What could possibly go wrong?
Desiree?
Yes, thank you for your talk. Fantastic. I've heard bits of it over the years, so it's great to see it evolving. I recently heard a talk by Azza Raskin about AI to communicate, to understand communication, using AI to understand communication, for us to be able to communicate with animals. And it was phenomenal and terrifying at the same time. Mm-hmm. And I wanted to ask you to speak a little bit about the ethics, possibly of, I guess, first encounter in a Star Trekian sort of way, in which these new animals are released into the world and are communicating. And it's early stages in thinking about that, but I wanted to know your thoughts on that.
I think about all those things, and I hope I live long enough to see them. So AI is another one of my interests, and it actually used to be called machine learning when it was mundane and in the lab. But now that it's out and it's called AI, it's much more sophisticated. And you can tell I have a fondness for what it can do. I'm sitting back and watching right now because I had no idea that it was going to take hold like this. I had no idea. It's coming too fast for me to think about specific examples. I think that's all I can do right now. But I do believe that it will have some impact. It's already having impact on the growth of iPS cells, the analysis of iPS cells. It will almost certainly be involved in generation of gametes. I mean, we're all in lab now, but AI is really taking over a lot of the judgment that we had to train people for 10 years to be able to do. So it's creeping up, and once it creeps up, it jumps to the next thing. So wish I could say something concrete, but I can't get my brain around it.
Yes, Melissa?
Just a quick question, maybe just to bring it to a practical level for those of us in the audience that are not doctors or scientists and don't even play one on TV. What, related to your work, can we do as members of the public to help it along? And what's, in one sentence or maybe two, the reason that we would do that?
Yeah. So my reason for putting as much energy as I did, my main reason for putting as much energy as I did into this particular program with the rhinos is that I think we owe those animals that have been, especially the rhino is such a perfect example of it. It was killed because of its horns, which do nothing. But they had to kill the whole animal to cut off the horn. So it's really our fault. And this is, I've said this before, and I know it's a little obscure, but in that reserve in Kenya, there are armed guards with machine guns that protect the last two. And I don't have a machine gun, I'm not an armed guard, but I do have stem cell expertise. And so that's what I'm doing to save those animals.
Let's thank Dr. Loring for a wonderful talk.